Hello everyone. So today let's talk about pentose phosphate pathway disorder. But before that, what is a pentose phosphate pathway? It is an anabolic pathway of a glucose metabolism. Pentose phosphate pathway comprises of two branches, oxidative and non-oxidative. Reactions in oxidative branch are irreversible, whereas reactions in non-oxidative are fully reversible. The oxidative branch primarily depends on glucose phosphate dehydrogenase, whereas transaldolase is the rate-limiting enzyme for non-oxidative branch. So basically, pentose phosphate pathway is an anabolic pathway that utilizes the 6 carbon of glucose to generate 5 carbons sugar and reducing equivalent. And at certain condition, glucose can be completely oxidized into carbon dioxide and water. Now, let's take a look at the functions of pentose phosphate pathway. The first one is the formation of ribose 5-phosphate R5P for the synthesis of nucleotides, RNA and DNA, supporting cell growth and proliferation. Next, the formation of NADPH is for biosynthetic reaction.
Now I'm going to talk about the disorders of pentose phosphate pathway. There are at least 3 inborn errors in the pentose phosphate pathway that have been identified, and the most common genetic defect in human is the mutation in G6PD causing G6PD deficiency. So what is G6PD? G6PD is actually cytosolic enzyme that catalyzes the chemical reaction in order to maintain the levels of coenzyme NADPH, whereas for NADPH, it in turn maintains the level of glutathione in the cell in order to protect the red blood cell against oxidative damage. G6PD deficiency is actually an X-linked hereditary genetic defect due to mutation in the G6PD gene. These may cause a single base change leading to amino acid substitution, which causes functional variants in many biological, biochemical, and clinical phenotypes. In general, G6PD deficiency, there is a defect in the first oxidative branch of pentose phosphate pathway, and this pathway is irreversible. As a consequence, the NADPH production is decreased, making the red blood cell vulnerable to oxidative stress. And the most frequent clinical manifestation of G6PD deficiency are neonatal jaundice and acute hemolytic anemia, which is usually triggered by exogenous agent. However, G6PD deficiency also have its protective effects in our body. It helps us to fight against malaria and also coronary artery diseases, which has been attributed to the involvement of NADPH in lipid biosynthesis and activities of NADPH oxidase. In short, the most effective management of G6PD deficiency is to prevent the hemolysis by avoiding oxidative stress. Screening programs for the disorders are undertaken depending on the prevalence of G6PD deficiency in a particular community.
Another pentose phosphate pathway disorder is ribose 5-phosphate isomerase deficiency, also known as RPI deficiency. Only one case of this disorder has been recorded in history, making it the rarest disease in the world. The enzyme which causes RPI deficiency is the ribose 5-phosphate isomerase. It helps in the conversion of ribose 5-phosphate into another chemical, ribosyl 5-phosphate. This conversion helps the body to in turn convert glucose into adenosine triphosphate, ATP, which is a form of energy that is readily used by the cells to carry out their functions effectively. Besides, the conversion reaction also helps in the protection of the body from an unavoidable and harmful chemical process called oxidative stress. Ribose 5-phosphate isomerase exists as two distinct protein forms known as RIPIA and RIPIB. Both of it catalyze the same reaction, and most organisms express one or both of these enzymes. So, What is ribose 5-phosphate isomerase deficiency and its cause? Certain environmental factors at times change the positions of the DNA molecules in the chromosomes. Such as event is technically called mutation. When mutation occurs in the RPI gene from which ribose 5-phosphate isomerase enzyme is produced, RPI deficiency is caused. The exact mechanism of how less production of this vital enzyme occurs because of mutation is not understood yet. There is only one patient diagnosed of this disease in medical history. This patient was a boy and the diagnosis was made in 1984 through magnetic resonance imaging (MRI) technique. As a consequence of the RPI disorder, a brain disease called leukoencephalopathy is developed. Leukoencephalopathy is vanishing white matter is a progressive disorder that mainly affects central nervous system. This disorder causes deterioration of the central nervous system's white matter, which consists of nerve fibers covered by myelin. This disease generally occurs in infants and children. The symptoms of the disease is affected children may have slightly delayed development of motor skills such as crawling or walking, abnormal muscle stiffness, and difficulty with coordinating movements. For affected females, they may have abnormal development of the ovaries. Ribose 5-phosphate isomerase deficiency can be diagnosed by analysis of sugars and polyols in a random urine sample. Urinary ribitol and arabitol as well as D-xylulose and ribose are highly elevated. Extremely high concentrations of these pentitols are also found in cerebrospinal fluid. Unfortunately, no treatment for this defect is available and the prognosis also is unclear.
Transaldolase disorder. Transaldolase is an enzyme of the non-oxidative phase of the pentose phosphate pathway. It is encoded by TALDO1 gene. What does transaldolase do? Transaldolase abstracts 3 carbons from pseudoheptulose 7-phosphate and adds it in glyceraldehyde 3-phosphate. The resulting products are fructose 6-phosphate and erythrose 4-phosphate. Transaldolase deficiency. Transaldolase deficiency is a condition where abnormally low level of TAL enzyme. TAL deficiency will increase the accumulations of pseudoheptulose 7-phosphate and the failure to recycle ribose 5-phosphate through the non-oxidative branch. It is mostly happen in erythrocytes and lymphoblasts. This is due to the deletion in 3 base pairs in the TALD1 gene. Deficiency of TAL enzyme will cause liver cirrhosis, hepatosplenomegaly, and infertility in men. Signs and symptoms of TAL deficiency. First, coagulopathy, which is bleeding disorder. Second, thrombocythemia, decrease of platelet. Third, hepatosplenomegaly, which is enlargement of liver.
As conclusion, there are 3 types of common disorder of pentose phosphate pathway. However, glucose 6-phosphate dehydrogenase deficiency is the most common one, while ribose 5-phosphate isomerase deficiency and transaldolase deficiency are rare but still considered common in genetic field. Thank you.